some parts that or some issues which are relevant for you to get some feeling about it. So first of all, I don't have to recapitulate the biochemistry 101. Everybody of you should know about the fluid mosaic model for the structure of cellular membranes, which is more or less stating that our lipid bilayer membranes are transversed by integral membrane proteins, here shown in red as a potato model. They are touched some peripheral membrane proteins, here shown like this one maybe. There are also some membrane proteins modified, and there are also a lot of other modifications. So that is the standard view as we have it for membranes. So coming to component number one. Component number one are the lipids. Lipids are, from a physical-chemical point, a really difficult class of compounds. Lipids are defined as all kind of compounds which are soluble in organic solvents. So they are structurally highly diverse. So we see here a general lipid, with a head group and some which is polar, and some more or less hydrophobic appendix, acyl chain or something larger whatsoever. But it could be also a protein-like lipid. Come to this later. So these kind of lipids can form different kind of phases. So they can be either in a soluble form, that depends on the so-called critical micellar concentration, CMC, which is the maximal concentration where I have the lipid molecules or the detergent molecules only as residing as a monomer. At concentrations above the CMC, they start to aggregate. They form micelles, bilayers, vesicles, liposomes, or even reversed micelles, depending on the water content. So these are the aggregated forms of lipids. And the kind of what they are now really doing, whether they're for micelles or lipid bilayers and so on, it's pretty complex. So when we consider lipids as parts of our kinds of cells, they are hugely determining for the overall function of cells. They're also important for the kind of shapes which cells can adapt or can adopt. For example, lipids can be found, different lipids, for example, here at these sharp bands, of the thylakoid. That is part of the chloroplast, where we have this kind of granum and the thylakoids in it, and the grana formed by the thylakoids and with photosynthetic reaction centers in being part of these membranes. But we have corners here, sharp bands, and you can predict probably the lipids are different here than there. And maybe there are some proteins associated with these kind of lipids here in the bands than here in that range where we have the photoreaction centers and the light harvesting complexes residing. So the lipids are also very important for the function of our proteins and the location of our proteins to which we do belong. As I told you, lipids are highly diverse. You know that from the biochemistry 101. Here you see some excerpt. Besides the normal lipids like the glycerolipids, also here some more complex lipids like lipids which are highly modified by glycan chains. We see also lipids which are derived from isoprenoids, or we have here also compounds which are also lipids like the highly toxic aflatoxin B1, which is formed by fungi. For example, when you like your organic fruits and they start to rot, with fungi, the likelihood that you die from aflatoxin B1 is probably higher than for most pesticides, which you have to wash off anyway. But that is part of the problem of aflatoxin contamination. So I hope that I have not, it's actually there's a mistake, phosphatidyl lipids, yes. These are a class of lipids which are given by two acyl chains which are esterified to glycerol, which has here additional phosphate group ester to the glycerol, and here as part of the head group different compounds. In this case it's it's choline, and they are not only diverse in the head groups, the lipids, but they're also diverse in the kind of acyl groups, as you can see here. One and the same kind of type of lipid, like here, that is phosphatidylcholine. Phosphatidylcholine is also a summary of a lot of different lipids, which differ by having different kind of acyl chains incorporated, especially at the C2 position of the glycerol. They are highly diverse. Here they have mostly C16 palmitic acid, but at the C2 position they can be very different. For example, oleic acid, POPC, PLPC. You see, these are the subnames of phosphatidylcholine which do occur. And that also does mean that when we have membrane proteins, we mostly are asking ourselves when we study membrane proteins, in what context do we study them? Is it the context of the right lipids? The right lipids would usually be the kind of lipids from the host. So one of the major problems that we do come up with lipids, and of course, especially when we study membrane proteins, being part of lipidic phases, are the very difficult phase diagrams, the very complex diagrams we do observe. For example, for this super simple lipid, dipalmitoyl phosphatidylcholine, DPPC, here shown. That is a phase diagram. It forms the lamellar alpha phase at relatively high water contents. Here you see the number of waters versus the lipid. But when you decrease the water content, it starts up to make a different lamellar phase. And that's not the only point. You see also, or you notice also, we have here also, of course, a temperature dependence in it of the phases. That means when you come up from this kind of water content, you go here with rising temperatures. You pass several phase boundaries. So there's a huge temperature dependence on the characteristics of the lipid membrane, which is the lamellar phase. When it's forming a lamellar phase, it's changing upon temperature. That's pretty hard to predict, and when you consider now that actually in life we have mixtures of lipids existing, not only this single lipid, but also the other lipid types, not only one phosphatidylcholine, but also phosphatidylserine, cholesterol, and so on. The phase diagrams are the hell and become almost unpredictable in many ways. So here, just to remind you, something about the lipids or slash detergents, what they can form. So most lipids we are talking about belong to this class, which form the lamellar phases. Here, this L-alpha phase. In that case, the diameter of the head group is almost the same as the diameter of the hydrophobic appendix that we do have here. That can be different when you have a normal detergent, for example, in your washing powder. You have usually detergents or lipid-like compounds like that one. They have a much smaller diameter in the acyl group, the hydrophobic acyl group, than in the polar head group. For example, dodecyl sulfate, which some of you know now in the practical course, clearly belong to this type of lipids. So you see at high concentrations it can form either micelles, or here in this case it forms some hexagonal phases, at high concentration, yes, with a little bit of water maybe here in between, which give rise then to this kind of mixed phase of water and this kind of detergent. You can also have the other way around, of course. You have very large diameter here in the hydrophobic area, at least in the cross section, and here for the polar head group. And then you get an inverse hexagonal phase. They're also known for some lipids. But in nature, we usually have that kind of time, the lamellar phase. So here is some overview about the different kind of phases that we do observe. Actually, just for those ones interested, before with this deep palmitoyl phosphatidylcholine, the P was for rippled, yes, So lamellar means it's a plane. It's a plane. It's a plane. It's a plane. You know, the face is planar, but it can be also rippled. That's a different phase apparently that can go on. It depends, of course, on the exposure of the head groups, you know, to water and to counter ions. But we also have some other groups, some other phases here. We had just had the hexagonal phases, but there are also some which are called cubic phases. They are pretty cool, cubic phases. So they are also disordered. Yes, they are almost all disordered, like the lamellar phase. They're also fluid, but they are pretty distinct. These cubic phases do occur with some lipids. You see that here, the I is for cubic, which are here in the phase diagram, depending on the water and the temperature to be found. And some of these cubic phases have a particular space group, here IA3D, you know, so that is a kind of long distance order these kind of cubic lipid phases have, and some of these lipids can form them. Why are they so interesting for structural biology? Actually, this lipid here is monoglucose deacylglycerol. It's occurring in chloroplasts and has something to do with packaging in chloroplasts, for example, also of the protochlorophyllid oxidoreductase, as much as I do remember. And that is interesting. So humans don't have cubic phases and also not this kind of strange lipid from plants. But plants are not strange at all. So here again about the kind of what I already mentioned, you know, in words, you know, now here in numbers, yes, you know, in formulas about the relationship between the form of a lipid detergent and the phase it can adopt. You know, here's a value called P that can be calculated from the volume, from the length, from the average statistical length of the hydrophobic group and from the cross-section area of the polar head group, and giving you this kind of this order of this kind of different phases. So point is about this kind of order is, you know, lipid membranes of this kind of lipidic phases. They can be planar. We have said that already. L-alpha was planar. We have also learned that there are also rippled lamellar phases, which go like this. So they are not so different from the planar phases. That means, you know, they can have curvatures, local curvatures, which are either negative or positive. You see here, the curvature is 0 in the planar lamellar L-alpha phase. The curvature is calculated by the inverse of the apparent radius. So the radius would be somehow here, you know, that would be part of a circle, and they are put on this kind of head groups. So C1 and C2 can be calculated, you know, for a phase, and there is a general point, you know, that of course they have to counterbalance themselves, and you see also here you need some energy to distort the planar membrane, yes, you know, according to some kind of force constants, yes, you have here. So in order to change the shape of a lipid membrane, you have to invest some free energy, and that's depending on the characteristics of its components, and that can be at least simulated or modeled. For example, molecular dynamics, it can be well simulated. Okay, that I think we have already talked. That is redundant, this slide. Coming back now to the cubic phase. The cubic lipidic phase I have just mentioned before is interesting because the curvature here C1 and C2, there are 0. That is the boundary condition for every minimal surface that is being formed, because that is the way where delta G adopts a minimal value for the distortion. So in this case, everything is highly curved, as you do see. So of course, the C1 is always counterbalanced by the curving at the other side around of the bilayer. So it can be transformed from the kind of planar bilayer to this kind of phase. Now, this kind of cubic phase has an interesting characteristic. So, whereas a lamellar phase separates the aqueous space into two different compartments, just remind here, two different compartments. A lamellar phase is usually always separating our bulk surface, bulk water, for example, in two separate compartments. Here, this one is inside, this one is outside. What is in cannot get out without crossing the membrane. What is out cannot get in without crossing the membrane. It's a bit different in the cubic lipidic phases. So here, when you are a membrane protein and you are incorporated here, you can diffuse through the whole three-dimensional space. You're not restricted like in a lamellar phase to 2 dimensions. And the other point is, so that is interesting. So usually when you have membrane proteins purified and you incorporate them into lipidic bilayers and you centrifuge them down, you have then stacked bilayers, and the membrane proteins, they cannot go from one bilayer to another one. They have to stick in their own one. So even when you have a greasy mass at the end that looks more or less transparent and homogeneous, it is not. So it is it's restricted in terms of movement. Here in the cubic lipidic phase, the membrane proteins can freely diffuse through the whole phase. And the other point is, you will see here, these are the aqueous spaces. One is shown in blue and the other one is in green. So in the ideal cubic lipidic phase, these two kind of watery compartments do not come into contact in the ideal case, because there's always this lipid in between. But nevertheless, everything that is incorporated in the membrane can freely diffuse through the cubic lipidic phase. You have also some other phase of cubic lipidic phases, which are quite known. The IM3M phase was the one where one was first able to crystallize membrane proteins in it. This method was introduced in the year 1997 by the group of Ehud Landau at the Biocenter Basel. Now we also have some other lipidic phases which can be also used, depends a bit on the lipid itself. For crystallization of membrane proteins, we can now use this kind of cubic lipidic phases. So we have just to see that we somehow incorporate the membrane protein into this kind of cubic phases, which is some kind of method, that can be relatively easy adopted to the lab. There are also some other phases, like this one here, this kind of sponge-like L3 phase, that it's done with a different kind of lipid. As you can see, a sponge is not something that is very well ordered. You see this here, L3 phase. It is, of course, not ordered, So there's some local order, but not far order. This has some advantages. It's a higher water content, the sponge phase, but has also the other, and there's no clear separation between the watery channels because it's really a sponge, it's really not well ordered. And you can also easily prepare that. So there are even commercial crystallization screens available to do the crystallization of membrane proteins in these kind of phases, in these sponge-like phases. So that was about lipid membranes and the kind of structural characteristics, and also some of the purpose to be kept in mind when dealing with embedded membrane proteins. Let's come up to some of the functional characteristics of lipid membranes. So first of all, when you consider the issue, what is pressure? Pressure is force per area. So clearly we have here 1 bar, but what is the pressure inside a lipidic phase? What is the pressure I have when I'm embedded here in this lamellar phase? What is the pressure of a membrane protein that it's gets being exposed to? Trivially, you may say, it is maybe 1 bar, like in the water when I'm a soluble protein, and here in the soluble bulk phase, I have a pressure of 1 bar, at least here on sea level. But that is not true in the membrane itself, because in the membrane, all these head groups do electrostatically repulse each other because they are negatively charged. There are clearly also some counter ions here present, some magnesium and calcium and sodium ions and whatsoever. But there's some repulsion here, the head group repulsion. And there is some attractive motion or some attractive force here between the acyl chains. So the pressure that I do experience inside a membrane is not homogeneous at all. So that has some effect on everything that gets incorporated into a lipidic membrane. So when you are a membrane protein and you want to change your conformation in order, for example, to do some transport, I think we get one talk about transporters, you have some conformational degree of freedom that you can move yourself, your structural elements. That can be, of course, depends on the composition of your lipidic environment. So you see this here, somehow highlighted for membrane protein, yes, somehow here the pressure it exerts depends on the position, and of course you can change the kind of function of the protein by changing the membrane pressure. For example, by changing the composition, which would make you, for example, your membrane protein non-functional at a certain kind of composition. Also, structurally, it would look intact. So the lateral pressure inside membranes can be pretty high, as you see here, about 400 bar or atmospheres. Oh, that was 400 bars or atmospheres. So membrane proteins are being let's say, controlled by the kind of environment. Let's see for an example. Let's take the major mechanosensitive channel MCSL. This is a channel that we do find in bacteria. I think MSCL is from Escherichia coli. And what is the purpose of this channel? First of all, it's a beautiful structure. Yes, I like structures. You could ask yourself, how does it look like? Okay, in red, we see here is one polypeptide chain highlighted. So as you can see, it's apparently a pentamer. A pentamer here, one helix, second, third, there's some element here, and here also some short helix. And this inner helix here forms with other helices from the 4 other monomers this kind of Aris-like constriction. So that's the view that we do have now from the top, when we would look from the top of the membrane. The membrane would now surround this mechanosensitive channel. So what is its purpose? So when bacteria are exposed to hypoosmolar stress, so for example, when it starts to rain and you are a bacterium, and before you have been in a pretty muddy place which you did like because it gave you a lot of nutrients and whatsoever, but suddenly everything gets within seconds diluted 100-fold, and you have suddenly a very strong osmolar pressure inside. What are you doing? Possibility number 1 would be just to blow up, and you are dead. Or possibility number 2, you have some emergency valve that opens up upon this and just releases a lot of anything from your cytosol till actually you have the same osmolytic pressure inside yourself, or at least an osmolytic pressure that can withstand the hyperosmolar condition that is surrounding you. And that is the purpose of the mechanosensitive channel MSCL. It opens up as a security valve. So there has been done a lot of studies on this protein. The structure, interestingly done here. I don't have this the simulation, unfortunately, here from Klaus Schulten's lab in Illinois, who did first studies on the structures, because the structures were the closed state. You see, this is the closed state. In this situation, nothing can go through the interior pore, where we have this 5 helices here arranged. And he was simulating what would happen When I have very strong hypoosmolar pressure, you can predict what that means. Also lowering of the lateral pressure inside the membrane, because you have this pressure from the inside that is pulling out everything. So the lateral pressure decreases strongly, and then you can observe a transition to an open state there where actually you suddenly form here a water-filled channel that allows water, but also of course a lot of osmolarity generating nutrients, glucose, ATP, whatsoever to escape in your surrounding. Clearly it's not very healthy for the bacteria, but it's an emergency valve to survive. So you see that here in detail. So here's a constriction site in this membrane protein channel that is closed, but upon mechanic stress, it gets opened up like an iris from a camera and opens up then to let the water-filled stuff then through. Okay, so here again in a bit more in diagrams, closed state, some intermediate state, it depends on the pressure inside the membrane that is being a result of the difference of the extracellular osmolarity and intracellular osmolarity, and of course of the characteristics of the lipids. But when it changes suddenly, it changes also the pressure profile here inside the membrane, and then the mechanosensitive channel can open up. So that is a very good example how important it is to have in mind when studying membrane proteins, at least those ones which have some function in transport or in conformational change or vesicle fusion and so on, to consider this. Another point is sometimes lipids are associated with a given membrane protein. In some other cases, they are not associated. So we have here on the left-hand side the mechanosensitive channel that we have seen before. Actually, as you may also here recognize in this simulation, or not recognize, at least it's not highlighted, you see there are a lot of lipids here in the simulation around it. It's a pretty computer-intense simulation that is done here. But none of the lipids has some preferential interaction site with the pentameric channel. For comparison, another membrane protein, the ammonia channel AMTB from Escherichia coli, that forms a trimer. Its function is to allow conduction of ammonia ions inside the E. coli cells because they require nitrogen. So this transporter is highly specific for a single lipid species, for phosphatidylglycerol. So this one is almost permanently attached here in the interface site, so the PG is stabilizing the trimer. So that is very often found that some lipids are contributing to the stability of a membrane protein and sometimes also to its function. I give you an extreme examples are our mitochondria. Our mitochondria have an inner membrane with a respiratory chain, and there the lipid cardiolipin, which is unusual and only found, or almost only found in the inner membrane of mitochondria, is very often to be found to be associated with these very large machineries which build up the respiratory chain. Okay, so when you like to measure pressures, you can do this also experimentally, at least to some extent. Simulations are nice, but experiments are nicer. I hope that in physical chemistry you heard about the Langmuir blodgett balance to measure this kind of interfacial pressures, but otherwise I explained to you, you spread a lipid monolayer on a water surface. The lipid monolayer orients spontaneously its head groups into the water and its hydrophobic side chains into the air, because air is hydrophobic, of course, yes, and then you have here this balance, here's this lipid spread, and then you have some mobile barrier and you can move it, and then you can measure the pressure that is being exerted on this mobile barrier. You can apply pressure to make this smaller and smaller, and then you get this kind of curve here in nano per nanometer per molecule calculated, or nanometer per nanosquare meter per molecule calculated. So when you start here, it's a gaseous-like phase, so the individual lipids do not interact with each other on the water surface. You make the area smaller, then you come to a transition phase where they start then to condense to this monolayer. Then you have the monolayer formed, then you have to exert further pressure, now stronger pressure, to make it smaller and smaller. And somewhere then you get additional condensed phase here. So one can measure also this kind of pressures to some extent. So I think that's from the Biochemistry 101, actually a slide. Yes, it's just telling you that also temperature is one of the most important factors anyway, also when dealing with membrane proteins, because it has an effect on the kind of mobility of these lipids inside the lipidic membranes. So for having a functional membrane protein, you have to be in this phase, which is liquid crystalline-like, but you have free lateral diffusion. But when you're in a real lamellar phase, like in this L-alpha phase, which is highly ordered, you have no lamellar diffusion anymore occurring. So this is kind of the liquid crystalline-like state. That is actually the state which our cells usually do adopt at the temperature greater than the Tm. And they do adopt in many ways. So, for example, they can change the content of unsaturated fatty acids inside as part of the lipids. You see here temperature and the number or the percentage of having unsaturated fatty acids inside your lipid bilayer, and clearly it increases. So Escherichia coli, when growing, it can adapt to up to 12 degrees Celsius. We can grow it in our shaker flask, but clearly these bacteria are pretty different in terms of the membrane than those ones which grow at 37 degrees, because those ones don't need so much unsaturated fatty acids like those ones here at lower temperature. You see, saturation level increases with temperature. There are some other organisms existing which have also some very strange ideas what they do, yes, you know, to regulate the kind of melting temperature of the kind of plasma membranes. For example, this strange bacterium Acoloplasma laivadi, which I got from this fancy textbook from Lilius et al. He has some very funny examples sometimes. I didn't know this kind of bacterium before, but at least the point is interesting. One cannot only change the fatty acid chain, one can also change the head group and modify by this the melting temperature of the lipid bilayer. As one can also see here, you know, where one can do experiments. That is actually not from this one. Actually, I think it's the wrong slide. I put the wrong slide in, but they showed that one can do this with this kind of lipids. So now I have talked quite a bit of time about lipids. It's actually taking a lot. I could now take a lot of time into talking about how lipid membranes do fuse. We would need probably quite a lot of time to talk about it. It's a non-trivial process. So when you have vesicles or cellular compartments, here shown as red circles, and this here just a little part here of the vesicle, they are not fusing spontaneously. That is not happening at all. They are not spontaneously fusing, because in order to fuse, they have to form some fusion pore here, actually, where the kind of polar side chains go to the side in both of these neighboring lipid bilayer membranes, so that the hydrophobic groups can go into contact. And then, only then, when this kind of transition pore has been formed, then it can start to grow more. You see this here on in the overview, and then finally it can split off again. Here, so it's a pretty complicated process in terms of the topology, and then you have two vesicles fused. So that is usually not happening spontaneously. I will give you not to talk about this. It would be some part for molecular cell biology, but also for structural biology. We have proteins which are actually binding to the lipid membranes and affect their kind of local structure, and which are for acting as catalysts for membrane vesicle fusion, and they are important factors of vesicle formation, of budding, and actually we also start now just recently to work on this a little bit, because has been some interest now how this kind of vesicle transport machinery has evolved in eukaryotic organisms, because many part of the proteinaceous machinery has been discovered in archaea, and also we have something discovered that may be also relevant for this process. But in common with the previous talk, think about it, also the lipids which have to accumulate here in this kind of transition pores, they have to follow up in terms of the geometry, the requirements of this. So that one fits well, that one doesn't fit well. So you can also predict that also these proteins may have some effect of lipid localization. Maybe the final thing about lipid localization. Natural membranes are super complex in terms of the composition. Told you that. So here you see some excerpt, that looks more like you have it in a usual eukaryotic membrane. So where cholesterol is, for example, being found a relatively high concentration on one of the leaflets, on the outer leaflet, on the outer leaflet of this membrane, a relatively high concentration of cholesterol. So Kai Simon and his friends, they proposed in the 90s or end of the 80s a new hypothesis what this may be good for, because they found when they purify certain signaling proteins, remember about receptor tyrosine kinases and the other signaling proteins we have talked, when they purified them from the membrane, they very often found that they stick together with other signaling proteins, and actually that these kind of sticky preparations are enriched in cholesterol, in this kind of steroidal compound. So they formed a hypothesis, so-called lipid rafts, stating that in the membrane there is some phase separation occurring in addition to being a lamellar phase. You have inside the membrane now a phase separation where a subcompartment is formed that is enriched now in this kind of cholesterol in terms of ceramates, lipid type, also in terms of some proteins which are associated with these kind of proteins. And by this, we would have some local accumulation of functional components inside the membrane. So as you see this here, that can be also emulated, pure single lipids and glycerophospholipids, mix them together at 37 degrees, they mix, then you add some cholesterol to it, and then suddenly they start to unmix each other. And that can be actually being also done nicely in vitro with pure components, this kind of unmixing event. Here you see that a bit more in the scheme. You have at the beginning a more or less lamellar phase, okay, maybe more in the liquid crystalline state, so that we have mobility. You add the cholesterol, it starts to make a phase separation. That can be found. So there are some proteins known which are associating with this kind of so-called rafts. So for example, the flotillins, I think Peter Graumann did work till recently, or maybe still nowadays, on bacterial flotillins from Bacillus subtilis, because there's a question of how important are these kind of lipidic rafts? Do they occur also in bacteria or not? It is somehow under debate. And are they important for signal transduction or not? So it's an endless debate that is interesting. After about 30 years, is somehow found its solution somewhere. Yes, it has some influence, yes, and it's very complicated. I think that was the right prediction of this model. Here I have, very strangely, I have here a German slide. Oh, interesting. So what one can do to get some more structural information of lipid biology, one can do smaller angle X-ray scattering. That's a way to do so, or small angle neutron scattering to see some of the distribution curves, but I think I skip this now. The thickness of, I didn't translate those ones, the thickness of the lipidic membrane is about 30 angstroms, 3 nanometer. So that means when when people talk about how thick is a membrane, it's very difficult to give a clear answer. So the hydrophobic part, clearly 3 nanometer. With the head groups, a little bit more. Yes, you know, the interface here is also being about 3 nanometer. So you can up to even more. Here in this case, it's up to about 6 nanometers. So the water hydration starts here. So when you have membrane proteins incorporated, the apparent thickness can be more than these 4.5 nanometer that you may have when you just stick here to the position of the head groups. It, of course, can be then thicker than that. But the true hydrophobic core, 3 nanometer, not more. And thickness has some influence because the thickness depends on the length of your acyl groups that you do have here. That is relatively trivial. That was being also experimentally found by the group of Engelmann in the 80s. Just to show you that one can see full membranes. That is one of the first views that one could look on a complete membrane. Now, due to the increasing number of cryo-EM structures, we will get more of these kind of views. So you see here a bacteriophage. It's a virus that infects bacteria, PRD1, and that one has an outer protein shell, here shown in blue. And in the inside, it had high electron density. Here inside, that is the DNA, high density because of the phosphorus atoms, which are part of the DNA. And then here these thin layers, you know, which have also higher density. And these are also coming from the phosphorus group. So here's the layer for the one head group layer and the other head group layer that is made up by this lipid bilayer that we do have here. And people know what's actually out. And by this, one could also measure the apparent thickness of a real lipid membrane. From peak to peak, about 3 nanometer when going from here to there. So let's talk a bit more about our membrane proteins. After having talked quite a lot about the lipids, but I was thinking other people don't talk so much about lipids, and I don't know whether in your previous studies lipids played any major role. Interestingly, they play a major role, but nobody wants to work with them because they're very difficult to handle. That is also the case of the membrane proteins because they're in the lipidic environment. So membrane proteins can be classified to peripheral and to integral membrane proteins. So we talk only about integral membrane proteins. Peripheral membrane proteins are thus those ones that are stuck on the surface of the lipidic membrane. Some of them have a single helix that is half in the membrane, half outside the membrane. For example, the cyclooxygenase, very important enzyme for the function of non-steroidal inflammation inhibitors, for example, like aspirin, but we are not talking about those. We are interested about integral membrane proteins, mostly about those ones which are not simply just anchored to the lipidic membrane by some lipid anchor. Actually, we work on some GPI anchored proteins or GPI anchoring in our group currently, but more on the transmembrane helices. So these are the transmembrane proteins which are really going through the whole membrane. So there are two types already at this position. One just requires 23, 24 amino acids to being an integral transmembrane protein. It has just a single transmembrane helix that is going through. And those ones which have more complex structures inside the membrane. So we are more interested for today's purpose more on those ones which have multiple passes going through the membrane of their polypeptide chain. So there are of these kind of multi-pass membrane proteins. There are two major classes, two major types. One of them is just using alpha helical transmembrane helices, abbreviated TM. You see that here we have seen already bacterial rhodopsin in one of my last talks that had 7 transmembrane helices here. That is probably something very similar. It also has 7 transmembrane helices, but it could be also 4 or 5 or 12. Many transporters have 12, 10, whatsoever. So the alpha helical type of integral membrane proteins, they have 0.1 different number of transmembrane helices, and the arrangement of these transmembrane helices can be highly diverse. So here it looks like that they are more or less going perpendicularly through the membrane, but the angle they can take is highly diverse. They can also have different arrangements relative to each other. So that means when you compare one 7-transmembrane helix protein from one family, for example, bacterodopsins, we had it last time, with another 7-transmembrane helix protein like rhodopsin or this G protein coupled receptors. On the first, they have the same number of helices, but when you look on the structures, the arrangement of the helices is somehow different. It's not easy to see, how actually one could transform the one packing to the another packing of these two types, and that's true for these alpha helical membrane proteins in general. That is also a reason why it's so difficult to say how many different kind of folds do exist for this class of membrane proteins. Because when I have the same number of helices in this 7, is it really a clearly distinct fold, or are there also existing some in-between candidates that are somewhere in the transition between these two extremes? It's pretty difficult to get this reconciled. So different arrangements, but many structures are formed, and membrane proteins are forming up about a quarter of the proteomes. So a quarter of all proteins are membrane proteins. Well, there's a problem, of course. Alpha helical membrane proteins do occur in the plasma membranes, their main occurrence, of course. They also occur in the inner membranes of gram-negative bacteria, okay, because they are plasma membrane. The inner membrane is the plasma membrane of bacteria. And they also do occur in the inner membranes of chloroplasts and mitochondria. They also occur, of course, in the membranes of peroxisomes, of endoplasmatic reticulum, Golgi apparatus, name me something, and I probably will tell you, yes, they do occur there. So they are the predominant class of membrane proteins, at least in terms of the occurrence. The second type of membrane proteins are the beta-barrel proteins. The beta-barrel proteins depend simply on the fact that here we have a beta-strand architecture, a beta-strand derived architecture, where the strands close to a beta-barrel. Remember the introduction about protein domains. A barrel is given when the beta sandwich, a beta sheet is closing itself, by making the antiparallel interactions with the last strand of its sheet. Then actually it's enwrapped. So this is a beta barrels kind of structure, or porine-like structural type. Unlike those ones before, with the transmembrane alpha helices, this kind of structural type is pretty uniform. First of all, in almost all, but there are some few exceptions about that later, we have an even number of beta strands. That makes sense because remember, antiparallel beta strands, up, down, up, down, up, down. So the repetition unit is 2, up, down. You can connect these kind of beta hairpins, 2-fold, 3-fold, 4-fold, 5-fold, 6-fold, 7-fold, 8-fold, and of course the number of beta strands you have is even. So they occur on the outer membrane of gram-negative bacteria, the outer membrane, so that is outside, peptidoglycan layer, inner membrane, and on the outer membranes of chloroplasts and mitochondria, but nowhere else. So that's different for them. That does not mean, that there are not sometimes alpha helical proteins in some of these compartments, but they are very rare. Mostly it's this kind of beta barrel-like type. So what does it mean in structural terms, or in terms that is more related to the structure? So first of all, what about the transmembrane helices? It's a normal helix, normal alpha helix that can be distorted at some position maybe, that is right, by a beta bulge or by a p bulge or a 310 helix. Usually it's an alpha helix. So the length is in the range of about 24 amino acids. That is a typical length of a transmembrane helix. But it can be also longer, because when you are not crossing the membrane perpendicular, but highly inclined to the membrane normal, then of course you need more amino acids to cross the membrane. Then you can require maybe 30 or 32 amino acids to come through the membrane. So the minimal size number of residues is about 20 to come through. So this is the average number, 24. The median is about 25. Compare this with soluble proteins. When you have a soluble protein, the average length of an alpha helix is 15. So shorter. In general, they are shorter. That doesn't mean that we don't have very long helices in soluble proteins, sometimes very, very long alpha helices, but on average 15. Second point is, when we have a multi-span membrane protein, these kind of transmembrane helices make up not everything, because we have loops connecting them with each other, sometimes even domains connecting them with each other, or some larger spans connecting them in the cytosolic or in the non-cytosolic environment. So that means they make up about the half of a transmembrane protein. Remember, a protein like maybe rhodopsin, what has it? I guess about 340 amino acids, will have about 170 amino acids, which are in the 7 transmembrane helices, given this kind of rule of thumb. The next point is, they often exert conformational flexibility, because when you remember, they are somehow paired with each other, they are associating via the hydrophobic chains. But that is not a rigid structure. They can move relative to each other. At least those ones have to move, which are forming transport, which have to change their conformation, or which are as part of a signaling protein like rhodopsin or G protein coupled receptor. They have to move, they are able to move the kind of helical segments in the membrane, depending on the state in which they are. So conformational flexibility is possible here. Then the composition, mostly they just consist of hydrophobic amino acids, but there are exceptions. Exceptions are mostly, remember about last time, bacteriorhodopsin, residues which have some functional role. Maybe a charged residue is inside the membrane, but then it has a specific biological function in most cases, almost all cases. So apart from being uniformly hydrophobic, that does not mean that all kind of hydrophobic amino acids occur in the same position. So big aromatic residues occur more here at the ends of the transmembrane helix. Smaller and branched hydrophobic amino acids like leucines and isoleucines occur, or alanines do occur more here in the middle segments of the transmembrane helices. So that has something to do that the aromatic residues often do interact already with the head groups, and are already exposed partly to the more water-like environment of the head groups. A very good point about transmembrane helices is that we can predict them very well. So the hit rate in prediction, when I have a sequence, is in the range of 95 to 98% hit rate. So when you have a membrane protein, a sequence of a membrane protein, you can be damn sure that you can predict the number of transmembrane helices inside this protein. Apart maybe when you have highly inclined transmembrane helices, because then maybe they are so long that you would erroneously predict that there would be 2 helices, whereas there's only 1. But you have a 95 to 98% hit rate. Interestingly, here's TM-HMM, for example, given as one of the standard programs that we are using them. Interestingly, one can also produce false positive predictions. So there are sometimes soluble proteins which are predicted to have a transmembrane helix, but they don't have. For example, myoglobin is a soluble protein, but TM-HMM predicted in the beginning it would have a transmembrane span because it has a long helix, which is very hydrophobic. So what about the beta barrels? In contrast, so now we're looking here. When looking here on the projection, we see in the alpha helix, there's no space to fit anything in. A alpha helix is dense. It's not the case with a beta barrel. It can have space, and that means it can be water filled. For example, in real porins, they are water filled. They form water filled channels. Not necessarily. It can be also dense here, but it can. Second point. This structure is highly stabilized by this kind of beta strand architecture. So there's no conformation flexibility possible in general. So it will not open up spontaneously here the beta barrel to do some conformational change and do this and that, for signaling or for allowing some substance to cross. That is mostly not the case with beta barrel-like proteins. They are rigid. Mostly. There's one exception. Next point is, the amino acid composition is almost indistinguishable from soluble proteins. That means this is the hell. You have a protein sequence, and maybe in the absence of additional information, for example, like the presence of the signal sequence, you do not know, is it now, could it be that this protein may be somehow anchored in the membrane as a beta barrel protein? These cases do occur. And it's very hard to predict this from the sequence because due to the architecture of these proteins, many of them are alternating hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic. So they look like inverted soluble proteins where the hydrophobic groups look outside of this fold and hydrophilic residues look inside to this fold. Okay, and then they are also mostly water filled. So nevertheless, that means in terms of the amino acid composition, no clear preference for hydrophobic residues as we have seen it here before. Hard to predict. So let's make a look on some types of these kind of proteins. And actually we worked a little bit in the past of some of these proteins, but they are anyway very prominent, so not too unusual. So let's take the prominent cases from Escherichia coli. OMP-F is the kind of, let's say, the model for most of the porins. It has 16 antiparallel beta strands, but forms a trimer. So 1, 2, 3, it forms a trimer. This is a monomer. This is a monomer. Everything is antiparallel. Because it forms a trimer, it does also look a bit more complicated. You see, one side is higher than the other one. This side here is actually the side of the beta barrel where they do form the trimer interaction. Actually here with the strand-strand interaction between the different protomers of the trimer. Very stable trimer. It's surviving even an SDS page. It migrates in SDS page as a trimer unless you boil the sample up. Then it migrates as a monomer. So apart from that, we have here some loops. The function is mostly to let metabolites passing through. Everything up to a molecular mass of about 600 Dalton can pass here through. It can also pass, it's a bit selective for cations, but only very weakly selective. So anions can also pass. And there's here some restriction site shown here in red. Here is this green part. That is actually the way how it's making itself smaller in terms of the diameter of compounds that can pass through. It's controlling the kind of size range the compounds can have. That's, for example, very important, not allowing some very large antibiotics to pass this kind of channel without any problem, or even some small toxic proteins from some other nasty organisms. So this is making it is restricting the channel at this position. So for contrast, another protein, from Escherichia coli, it's the outer membrane protein G. It's actually not found. Mostly not expressed by most Escherichia coli strains. The gene is present on the genome. It's apparently getting only active under kind of some stress conditions. It's a monomer. It's very simple. You can produce it in gram amounts. So we produce this protein in very large quantities for a lot of chemical for chemical work. It's pretty stable. You see this protein 75 degrees Celsius in detergents, 105 degrees Celsius in 2D crystals. That is not unusual for beta barrel proteins. Many beta barrel proteins, because of this rigid architecture, are very stable, which cannot be said about the alpha helical one. Okay, and there are even some porins existing which are not which are not water filled, at least not in the normal state, like this protein here, FAC called. It's a transporter for the iron citrate complex. So somehow the bacteria have also to take up iron from the environment, and very often iron is well complex to citrate, as you should remember as chemists, that citrate is a very good complexant of a metal like iron. And interestingly also, this is a transporter for the iron citrate complex. You see, this is plugged. Yes, you know, this is filled. So there's no free passageway existing. And I have to look into the literature whether now in the year 2020 it's now glass clear how the iron citrate complex is getting through this complex. Clearly one can predict in order to get through the channel or through this kind of transporter, it has to do some conformational change, making it that to a transporter, which is partly unplugging here the porin-like interior so that the iron citrate can pass. But the pathway, at least to my knowledge, was not known, at least a few years ago. I have to make a look, or maybe one of you can make a look whether anything is new now new about the iron citrate transporter. Coming to some unusual protein, which is very important because our mitochondria depend on it. All mitochondria depend on it. Mitochondria have an outer membrane as this kind of gram-negative bacteria. So this outer membrane has to be permeable for compounds, for ATP, ADP, for NADH, it has to be permeable. For the metabolites, acetyl-CoA, for this and that, you know, everything that gets into the mitochondrion or out of the mitochondrion has at the end to pass the outer membrane of the mitochondrion. So the role of this protein is to make the holes in the outer membrane of the mitochondrion and to allow the passage of these compounds. It's called VDAC, very strangely called voltage-dependent anion channel 1, because the people found that it's weakly anion selective and that one can change the conductance by applying some electric field to the membrane. So what is interesting about this protein compared to the other ones I have shown you before, it's non-even, has a non-even number of beta strands. 19 beta strands are in it. So that is unusual. That means that strand 1 and strand 19 form a parallel interaction instead of the anti-parallel interaction that we usually have in the porin class of proteins. It's involved also in some reactions which are very important for the cells, for example in apoptosis, that means programmed cell death. And there it is thought that actually this kind of architecture is very helpful because this kind of parallel pairing of the beta strand is an inbuilt weakness in this beta barrel architecture of this protein. So there exist predictions, unfortunately not yet structures, that it can open up and accumulate and form interactions with other VDAC molecules or some apoptotic factors to open up very large channels in the outer membrane of the mitochondrium, so that the cytochrome C can escape to the cytosol from the inner membrane space and then start to trigger the caspase machinery. Very nasty reaction, of course, programmed cell death, but this protein has a weakness built in. Interestingly, it's not the only protein has this 19-stranded beta barrel architecture. There exists, of course, also another protein in the outer membranes of mitochondria, called the Tom complex, transporter of the outer membrane. It has almost the same structure, and that is involved in transporting proteins from the cytosol through the outer membrane, either to the inner membrane space or to the transporter of the inner membrane, to the TIM complex. So that has also a built-in weakness by being uneven stranded. As we have seen here before in OMP-F, here it's also partly plugged by an alpha helix, and that is actually the alpha helix that is actually playing a part here in this voltage-dependent closing and opening of this channel. So that about the kind of structures about outer membrane proteins and this kind of class here, this overview, they look almost very similar, you know. One has to add up some of those kind of proteins act really as membrane anchor. So there are proteins existing on the surface of bacteria which are attached to an anchor like this one and have then here projecting something out of the outer membrane. Here in many of these proteins which are attached by this to the outer membrane, the beta barrel has before functioned as an autotransporter. So these are proteins which also have, during their incorporation into the membrane, have allowed to pass some part of the polypeptide chain through. Then the outer membrane barrel has closed up, you know, and formed in the anchor, and that's it. Finally, you know how to classify them, these kind of outer membrane proteins. One can classify them according to their so-called shear number. It's giving us some idea how strong the inclination is of all these beta strands, whether they're going more like this or like that, how many strands are required. And the shear number is also some very simple geometrical descriptor of beta barrel proteins in the membrane, because what you're doing is you start from strand 1 and then you ask yourself from which residue of strand 1 you can actually go by a closed chain of hydrogen bonds, because we have antiparallel hydrogen bonding. Yes, it's an antiparallel beta sheet going back till you finally get here on strand 1 back. Here starting on residue L, residue K, going here back on residue L, and then the difference K minus L is in the shearing number. For example, the shearing number of 8 was part of some of these proteins that we had before. But beta barrel proteins have not been the first proteins which have been structurally resolved and have been understood in terms of the mechanism. So the first overall structure of any membrane protein that has been characterized, unfortunately, at least at the beginning, at low resolution, at a resolution of 7 angstrom, was made by Richard Henderson from the MRC in Cambridge, who used electron crystallography to get the structure of bacteriodopsin out. You see what he could achieve at that time. In 1979, I think, was the structure published. 1975, maybe I get, no, 1975, 1976. I have to look for the original paper, not for this damn textbook. So that is a model he got out. He could show as a first-time person, you know, that actually membrane proteins have this kind of transmembrane helix, because the density was clear enough to be interpreted as a helical element, and resolving some debate about the kind of structure about these proteins. Please don't forget, Richard Henderson got 3 years ago the Nobel Prize for chemistry with 2 other colleagues because of his work on getting now structures done by cryo-electron microscopy nowadays. So he worked, as you can imagine, from the beginning of the 70s at the MRC in Cambridge for more than 40 years to get cryo-electron microscopy working. So after this quick success with bacteriodopsin, he had to wait 30 more years till coming up with a methodology and with detectors which could actually fulfill what actually he always promised. So you see a bit more in detail the structure of bacteriodopsin. We talked about that last time. I won't go about so much in maybe a different aspect. As you see here on the right-hand slide, but also here on the left-hand, it's 1 monomer. Here are 3 monomers of bacteriodopsin. It's forming a trimer. This trimer is specifically associated with a specific sulfoglycolipid, from this archaeal organism, here shown in red, space filling, and that is filling the plug. That is acting like a plug, because otherwise you would have a hole in the trimere where nothing would be in it to fill also the trimere, so that nothing can go through it. So that forms the plug. Interestingly, only from one side of the bilayer, not from the other leaflet. So it's also a large lipid that is making the plug. The trimere itself is surrounded also by specifically interacting archaeal lipids. So you see them all here. They are all structurally resolved because they are part of the crystalline purple membrane. Each lipid has its specific position. These archaeal lipids, which are pretty unusual and make interactions in the interface and in between the bacteriolopsis monomers. That's the reason why this protein is forming a natural crystal, because it uses specific lipids to do this kind of aggregation. This one we have already seen last time. It's a simpler figure than the one I made a few years ago, but let's go to a different channel. Maybe you remember my Biochemistry 101 talk. It's a potassium channel, KSCA, also extremely important. First of all, it's again an oligomer. Now we have not a trimere like in Bacterodopsin, but a tetramere. But in this case, the tetramere is letting an open channel through which something can pass. Potassium ions can charge with a 10,000-fold higher conductancy for potassium than for sodium, so that we get up 100 million potassium ions per second passing the channel under saturating conditions. The protein itself is very trivial. You see the structure here is almost 2 transmembrane helices, and then there is a third helix attached to it that is not passing the full membrane. Here showing this one here, that is that is part of the selectivity gate, and a short extended strand-like structure, which is now here partly occluded and fourfold repeated. That is actually the selectivity gate itself. So that is a specific structural element. You see, you cannot predict this, yes, by just knowing I have 2 transmembrane helices and something attached to it. There are many other proteins that have 2 transmembrane helices, but they do not form tetrameres like that one. So the transmembrane helices are attached to this extended strand, and when you look to the extended strand, you see the functional part of this extended strand are the peptide groups themselves. So the peptide groups are projecting with the carbonyl oxygens to the center of the channel itself. So here, where we have this lineage of potassium ions, here is actually the fourfold symmetry axis passing through the tetrameric channel. So each of these ions is surrounded by a fourfold symmetric arrangement of these carbonyl groups. So 1, 2, 3, 4, and then also one from the back, because we miss the 2 strands from the back that we do not see, or one in front of us and the other one in back of us, because that is cut off now here away. So that is actually the central structural part of the so-called selectivity gate that is allowing only potassium ions to intrude into the site here itself. It's also interesting because when the channel is being cleaned in the full absence of potassium ions, actually, and then you would like to start to see whether it's conducting, it's not conducting, because it needs first to get some potassium ions first inside this selectivity gate to form this structure here in a proper state, and then to become also conducting. Besides the 4 potassium ions in the selectivity gate, there are 2 more. One of them are here at the entrance site, which is already waiting that it may come in. Yes, so it's loosely attached here to some of the side chains and the other parts. And one potassium ion is shown here that is fully hydrated by 8 water molecules. That is actually within the transmembrane region of the KCSA channel itself. So that is interesting because it means that after having passed the selectivity gate, the potassium ion finds itself in a fully hydrated environment back. Also, it has still not passed fully the transmembrane area itself. So when looking, what this means, the potassium channel allows almost passage of the ions without any kind of activation energy barrier. And the barrierless transport is, of course, required for the maximal transport rate that is faster than free diffusion of potassium ions in water. So we cannot have any kind of activation energy happening, and that is done by alternating the potassium ion with a water ion, to which it's of course coordinated with a potassium ion. So there's a file of potassium water going through this channel at the same time. So at maximal rate, this has then the 3 or 4 potassium ions within the channel, and you see here it's only a minor change it's doing from an 8-fold coordinated potassium ion, as seen here in this 1,3 arrangement, to this arrangement here. It just moved by less than an angstrom, maybe 0.7, 0.8 angstrom. It moved a little bit and is now 6 coordinated. And the energetic difference between 8 and 6 coordinated here is minimized to be close to 0. That is the reason why actually the potassium channel is selective. There are also some other channels existing, like the sodium ion channels, and these ones are much more complex than the one that we have seen before. I do remember when I was a student like yours, doing my practical Vertiefung in the Michel group, was my topic to purify this channel from some electric eel, some electric fish actually. Was very difficult to do so, but the structure was much more complex. It has a molecular mass of 250,000 Dalton. The other one was a very small protein, 30 kilodalton. The other protein actually comes in a similar form also in us as Shaker potassium channel. It's a bit larger, than I think 60, 70 kilodalton, but architecture is the same. But this one is really giant. So why is it so large, the sodium ion channel? First of all, it resembles KCSA. It's actually somehow tetrameric. You can see this unit, that unit, that unit, that unit are all repeating themselves. They are not identical, of course, but they are at least very similar. Then, second point, 4 of the helices form the channel area themselves by just forming an intramolecular tetramer. So this one, 1 to 4, is interacting with 1 to 4, 1 to 4, 1 to 4, yes, as seen before. And the helices 5, 6, called here, they are forming the voltage-dependent pedal that is actually changing its conformation in dependence of the applied electrical potential to the membrane. So this sodium ion channel is extremely important for us because we require it for nerve conductance. So you see here some of the compounds that are plugging this channel. They bind to the channel but plug the channel, like tetrodotoxin from fugu. So when you like to eat fugu in Japan, be aware tetrodotoxin is one of the most toxic toxins that we do know. It's actually interesting to see what it's derived from. You see all of these groups have a guanidinium group here. This one, like saxitoxin, that is from some dinoflagellate, causing some algal pest. Yes, when suddenly the seawater becomes toxic. Yes, dinoflagellates are a problem. Yes, when they make something like this, that causes then mass death of fish. Of course, because of the action on the sodium channel. Here see also the guanidinium group, same element. Here actually it's repeated again. And then also batrachotoxin from the frog of the Amazon Indians that is also working quite effectively in plugging this voltage channel. I will not talk about this voltage channel too much. It's a very complex structure. Actually, I was very impressed when I read as a student the papers from what was Cavendish, the name I'm a bit uncertain about it. when I read the papers as a student from him, about his work on the sodium channel, fascinating as it was, it was quite obvious that he also liked to crystallize. He worked over 30 years to get this protein finally crystallized and get resolved in structural details, how actually the potassium-dependent regulation of conductance works for this potassium channel. There are also other channels existing, like the chloride channel. We need chloride anions, clearly. Chloride channel here in this case from a bacterium. Actually, this is not a channel at all. Actually, this is a transporter, because this protein has a selectivity gate here. And when you see in the selectivity gate, here's the water apparently, there's no continuous passageway for a chloride ion to pass here from this arginine 147 to the other side to get in. So conformational change has to occur at this position. And when you look a bit carefully, you get the impression that somehow it looks a bit symmetrical at this position here, that the whole channel looks a little bit symmetrical in some way, and that is actually true. So it has some pseudo-symmetry inbuilt, and it requires conformation change, at least at the selectivity gate. So for this channel, we have actually a pseudo-symmetric arrangement of its two halves, or let's call it, it's called CLC channel, but let's not be too Pharisaic, to make a big discussion about that. So it has two pseudo-symmetric halves. So this one helix is pointing with its amino terminus towards the restriction site that we have seen here before with the amino terminus, in order to put enough positive dipole moment to the center of the transporter to stabilize the position of the chloride ion there. Okay, the structure of the chloride channel that I showed you was a closed state. I would have to look whether we have also now some open state available. In the potassium channel, the helices were coming from 4 subunits, also in the sodium channel similarly, and they point with the other end, with the anionic charge C-terminal end of the helices to the center to stabilize here the cationic charge that was formed by the potassium surrounded with the water in this kind of cavity. And here it could then escape then out and get into the cytosol. Finally, let's talk about, just shortly introduce the aquaporins. That we finish with the channels. The aquaporins are interesting. So the aquaporins discovered by Peter Ager, also Nobel Prize, like Roderick McKinnon, that was for the potassium channel, same year, for chemistry, is actually required for making our membranes truly water permeable. So when you're a red blood cell, you have a lot of aquaporin inside your membrane, that the changing osmolarity inside your cytosol, depending on the charging state of whatsoever you have in, can be quickly adapted. Interestingly, this is a tetrameric protein, but now, this time, the pore is not formed at the intervening site of the tetramer here, that is dense. It is formed by each subunit itself. You see here the pore being somehow highlighted. Here's a pore, there's a pore, there's a pore, there's a pore. The water conductance of aquaporins is extremely high. So when you would be able to have a paper sheet that would have aquaporins incorporated, or some let's say some not paper, of course, some lipid membrane, yes, which you would have densely packed with aquaporins, and that would be the end of your bucket, and you would start to throw on water on this bucket. You would never get it full, because it would get out almost immediately. That is the conductance of aquaporins, freely permeable to water. The reason why they are permeable is not so difficult to understand, because they have a continuous water channel, but there is one single point that is important. And the point is, but they let only pass water, but nothing else. Because when you have a line of water molecules, you can use a Grotius mechanism for water conductance to get actually protons conducted. Unfortunately, I don't have a slide, but maybe I switch shortly. So when you consider the water molecules, they are H bonded with them with each other. Clearly, they could just give here the proton from one to the next person by releasing it at the same time. Actually, that's not the best slide. Yes, for that purpose. By this way, the proton conductance is much higher than you would predict by diffusion of protons through water. So in order to avoid that actually continuous water chain would also conduct protons and would by this disrupt the proton motive force. That means the electrochemical potential along the membranes. This kind of water channel is disturbed at one particular position at this restriction site here, where actually two water molecules are off-rotated in such a way that they cannot form a hydrogen bond anymore. And by this little trick, aquaporins do avoid to allow proton conductance through the membrane. The restriction site is made by these NPA motifs, asparagine, proline, alanine, and they are actually also coming from two pseudosymmetric halves of the aquaporin. So two NPA motifs come close to each other at the restriction site, where actually the one water molecule is a bit off-rotated, the other water molecule is a bit off-rotated, and the proton chain is broken. Finally, about this, I will not go too much into the methodology. I think you should know membrane proteins are usually handled by getting them out of the lipidic membranes by using a detergent, by first disrupting the membranes with the detergent. So here it's soaking up the detergent, is breaking into pieces, and then finally with more detergent, when I add more and more detergent, I have then the membrane proteins incorporated in these micelles formed by the detergent besides of course micelles which have nothing incorporated. So these mixed micelles are actually the ones that we like to have, not those ones. And of course, you see, when purifying membrane proteins, the heterogeneity is a problem because sometimes they're not completely solubilized. There's some lipids attached. You have a lot of empty micelles or some micelles with some lipids in it, and to get a homogeneous preparation is pretty difficult. Here on the right-hand side, you see the kind of chemical diversity that is nowadays used in structural biology to purify membrane proteins, and this is a never-ending list of detergents that do come up with. Some of them are very cheap, like this one, like LDAO, costs just a few cents per gram, but others are very expensive. You buy a few €100 per gram, like digitonin, for example, this very complex compound that is very successfully used for some membrane protein complexes. Some of the other ones which are synthesized, especially on purpose for structural biology application or for membrane protein biochemistry. That makes membrane protein research to a pretty expensive bit of research. However, there are also other ways to handle your membrane protein nowadays, because biochemists spend money and membrane biochemists spend even more money. So there have been come up a solution to emulate nature a little bit with the trick that we use ourselves when we digest lipids. When we digest lipids, we add up bile acids in our gut, and then they do not form bicelles, they form this kind of nanodiscs. With our bile acids, they are pretty heterogeneous from the size, so they look a little bit like a lamellar membrane here. Yes, also our lipids in our digestion tract would look like that, but in order for handling here these membrane proteins, we have here a little synthetic protein around it that is forming here some oligomers around this kind of lipid membrane protein assembly. And this assembly is called a nanodisc because it's hydrophilic from the outside. That is done by this little synthetic protein. It comprises the lipids, which may be also our protein does really like, because remember, when I have something in my cellular environment, that is a bit very artificial compared to the environment in the intact membrane, remembering that one, what we have talked before. So here you have also a more native-like environment, and now you can study your membrane protein incorporated in nanodiscs by, you can try to crystallize it, but nowadays I think most people would prefer with nanodisc preparations of membrane proteins rather quickly to cryo-electron microscopy and to study them. So the way to make them is pretty similar like before. In the first part, you make a solubilization with detergents. They are not shown here anymore. They have been just omitted from this slide. So you purify somehow, you solubilize everything, and then you add up this MSP, this kind of fancy little protein that is forming then this kind of defined biset, and some lipids you have also to add up, yes, because then you can also make an assembly with defined lipids and not with some crude mixtures necessarily from your national preparation. But you can also, of course, make a phospholipid mixture. You can take also phospholipid mixtures to see what's more stable or not. So that is one of the ways how how we are now thinking nowadays about handling membrane proteins. And the number of membrane protein structures is increasing rapidly because of cryo-EM and because of the availability of additional new techniques like this one. So I will not give you an overview, or maybe when some people are interested to look what kind of membrane proteins are at the moment available. Actually, it's interesting. Now we have more than 1,000 unique membrane protein structures in the database. This one here, 3,000 entries overall, much more than before. Hartmut Michel from Frankfurt is not following up anymore, making this database. I think they have here some funding, in the US for going on with this kind of database handling and analysis. And now I would be potentially at the end. But I can make the Steve Jobs today again and say there's one more thing. One more thing. And hopefully you excuse me when taking a little bit of your time for one more thing. So we talked now a lot about membrane proteins and how they look like. But we could also ask, how do they get into the membrane? This is also not a trivial task. All membrane proteins are synthesized at ribosomes. We know. So first of all, it's a complex machinery to get a nascent membrane protein into its place and to get it being produced there. So you see here a ribosome that is translating some mRNA coding for a membrane protein, and this membrane protein actually has here in this example a signal sequence marked here in red. A signal sequence that is on the surface of the ribosome. When it comes up on the surface of the ribosome, the GDP-bound form of the signal recognition particle binds to the ribosome that stops also its translation at this moment. So there's no further peptide growth anymore happening. This arrested signal recognition particle ribosome complex undergoes an exchange of GDP to GDP, and now you see signal recognition particle is a small g protein, as we have seen it with P21-RAS. This complex, and is also structurally highly related to this class of proteins, same fold. This complex then gets being then translocated to the signal recognition particle receptor that is forming a complex with a channel called here the translocon in the closed state. So this signal recognition particle receptor as itself has a subunit that is a small g protein, is being bound in the GTP bound form. So the complex gets being formed of this and of that. The complex is formed. This diffuses off because the ribosome makes now a tight complex with the translocon. The translocon opens up. The other complex diffuses off and restores its components by hydrolysis of GTP to GDP, as you remember. and in the meantime, the translocon now, and the ribosome continues now with the translation, and during translation, everything else goes through this translocon into the ER lumen or wheresoever. So that is a general process of protein secretion and membrane protein incorporation. For secretion, the situation is very simple. So the protein, after the signal sequence has been recognized, it grows and grows, yes, into the ER lumen. And then here in the ER lumen, when it was to be a soluble protein, there's a protease called signal peptidase that positions specifically the signal sequence off, that stays here more or less in the membrane, and the soluble protein gets then somehow later its way to the secretory pathway, maybe up to the plasma membrane or outside being secreted. For example, like insulin is a classical example for this, when you are being asked, how is insulin formed? Yes, how is it secreted by the cells of our pancreas? So with membrane proteins, the situation is a bit different. They have to end up in the plasma membrane or in the lipidic membrane. To remind you, first of all, there are existing two types of membrane proteins. The one is called type 1, the other is type 2. Type 1, the N terminus is outside and the C terminus is in the cytoplasm. And I always say outside. Outside does not mean it's extracellular. It could be also ER lumen, Golgi lumen, whatsoever. In type 2, it's just the other way around. Type 1 is much more often to be found. I think 95% of all membrane proteins belong to type 1 and a few percent only to type 2. And usually we have, and from proteins that we are interested in to talk about, we have not a single transmembrane span, but we have several one of them. But they can be type 1 or type 2, depending where the N terminus is located. So here the N terminus is located in the cytosol, and here the C terminus is located in the cytosol. But here the position of the N terminus is a cytosol. So that would belong to a type 2 membrane protein, because when you would cut here, that would correspond to this kind of type. So that's interesting. So we translocate something, and actually it has to get up both orientations. The protein is some overview here of the different kind of protein, but I just skip this quickly because it's just for simplicity. So how is a type 1 membrane protein synthesized? Also now first a sketch before we come to structure points. So when the ribosome is docked on the translocon, the signal sequence is sometimes clipped off. It needs a signal sequence that is required for ER localization. Then it synthesizes more, and then it synthesizes the so-called stop transfer anchor sequence. So this sequence is very hydrophobic, and it tells the translocon to undergo now a conformational change to open up to the site and to release now the protein into the membrane. Here in this example, it's a single transmembrane spanning protein, because here the ribosome also goes off. But when you have several transmembrane spans, the ribosome would still be associated. It would not go off. Only when there's a long stretch where nothing is associated to it, it could go off. So that would be the simple way for a type 1 membrane protein. And you see here also some post-translational modifications like glycosylation in the ER lumen. Interestingly, there's also type 3 existing. They miss the signal sequence. Also very rare, pretty rare actually, type 3 proteins. What about the type 2 proteins, which you mentioned? Some of them are very important. For example, the beta subunit of the sodium potassium ATPase. So in this case, we don't have a signal sequence in the normal way. We have here something like an internal signal sequence that is also recognized by the signal recognition particle receptor and guiding it through the membrane. But you see here in that case, because it's so long, it forms a hairpin. And the hairpin here, and the hairpin here, the end terms here, amino plus pin here, that's the hairpin that is still being already here formed, that just sticks out now. So that will not be changed. So mostly these are relatively short chains that are looking here inside the cytosol. And then the rest is translocated through the translocon and then somehow finally released, and then you end up with a type 2 membrane protein. So that's the principle. And with a multi-span protein, okay, then you have several steps. First, here type 1, a type 1 case, now a type 2 case, I think here. First signal anchor sequence, then first stop transfer sequence, zack, 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 and then it's never going off, not like this one here. That is a very bad prediction. I have to change the slides. That's partly wrong this nowadays. Because we know much more about the protein translocase. We have structures which can explain the mechanism of how proteins can be translocated through the YA loop. First of all, you have to consider nothing can go through the translocon or SEC1 or SEC-YAG complex, a part of the polypeptide chain. That is remarkable because you have 20 different amino acids. They look all different. But no ATP goes through, no sugar goes through, also not major amounts of cations go through the translocon. So it has somehow be able just to allow the passage of the polypeptide chain, but nothing of any kind of soluble compounds. So that is interesting requirement for the translocon. This is fulfilled by the translocon by having a restriction here called poring that is closing it. That is actually the closing site. Actually, it's additionally closed by a plug region that is actually sticking up. So the combination of both keeps it closed. So what happens now when the plug goes away? So when the plug goes away, one would expect that many small little things can pass through this kind of poring. But that does not happen because in order to go away, first the polypeptide chain has to be pressed through the poring by the continuous translation of the ribosome that is here on top of it, so that it's somehow pressing the polypeptide chain through. And the second point is the poring itself is not a rigid structure, it's comprising methionines. So it's full of methionine residues. So methionines are hydrophobic, but they are very flexible. They are greasy-like. They are somehow like a little greasy lipidic stuff here at this pore site. They can be pushed to the side chains. And that is interesting because the same principle has been used by the signal recognition particle receptor that we have seen here before to recognize the signal recognition sequence itself. It's also very hydrophobic. The signal recognition particle receptor has also a stretch of a lot of methionines which are greasy and hydrophobic, but they're not very specific for what kind of hydrophobic structure they like to recognize. It has to be somehow hydrophobic. So that is actually the trick here, to have this kind of methionine plug inside the SEC-YAG protein. And then, of course, the plug in addition to open it up. The SEC61 or SEC-YAG complex is extremely conserved. Eukaryotes and bacteria, they are not very different in terms of their structure requirements. Also, the organisms are widely off in evolution. So here you see the mechanism also more in a more, let's say, way for a protein that becomes a membrane protein now. Because when now becoming a membrane protein, it's not enough that I just pass through the translocon, but the translocon has also to open up. It has to open up in order to release the future transmembrane channel to the lipidic membrane. And we have here a weakness inbuilt. You see here this kind of pseudosymmetric arrangement, again, pseudosymmetric arrangement as we have seen it before, for the helices 1 to 5 and the helices 6 to 10. They are connected here in a hardware with each other. There's a hinge-like region assigned to it, this backside. But here to the side, they can open up. So they can release something that is in In the translocan at the moment, a helix formed here. When it's very hydrophobic, it can interact with the kind of this region here and causes it to open up and to release it to the lipidic bulk environment. And by this, transmembrane helix is released. This is very interesting, complex mechanism, because it's saying that is something that a conventional beta barrel protein would not be able to do. However, remember that ones with the uneven number of beta strands, they can do something similar. So that's another story, not for today. So that is a mechanism as we understand now in structured terms. In reality, we have now a cryo-electron tomography analysis of the full complexes existing, of the full machinery, and it's even more complex. So here we have the ribosome, the eukaryotic ribosome with the 60S and the 40S subunit. It's docked with the exit channel of the big subunit to the 661 complex, to the entrance here, where now the polypeptide goes through. But please see, there are additional components now here in this cryo-EET structures to be found. One component is called abbreviated OSD and the other is called TRAP. So maybe the simple thing first for TRAP. TRAP is the abbreviation for translocan associated protein complex. It assists somehow 661 depending on what kind of substrate is coming up here. Clearly it is pretty close to the hinge region and also assisting in the release of membrane proteins to the bulk. So that's one of the many probable regulatory functions it also has for this kind of complex. Another very large complex, the OST complex, has been also very recently structurally resolved in detail, is the oligosaccharide transferase. This is a protein complex that recognizes asparaginins as part of an NXT or NXS motif. These asparaginins, when they come close here now to the OST complex, get the kind of oligosaccharide glycan to make a true end-link glycan antenna attached to them. That is actually the way how actually all these kind of secreted proteins get being glycosylated. And the majority of secreted proteins indeed gets glycosylated because they have some NXST motif inside of them. So that about this machinery. Maybe to finish now with today's talk with some very badly resolved slide. I told you, you know, that the propensity of amino acids in transmembrane regions is different. It's not enough that they're hydrophobic. I just show you that biochemistry, not only structural biology, but really true biochemistry, really true membrane biochemistry, or really true biochemistry that you can do in the wet lab can be very helpful to answer your questions. Here we have an idea from Gunnar Heinen's lab how to quantify the propensity of different amino acid types that they like to being part of a transmembrane helix. He studied a protein, a very simple protein, that has a helical element that can be depending on a single amino acid exchange being either being incorporated into the helix or not being incorporated. Just a finding here that he found during some of his studies, that this is a helical element and this is a helical element where the composition of a single amino acid determines whether it goes into the membrane or not. And he was doing something very similar. He made a lot of mutants at all these kind of positions. Or his co-workers, of course, sorry for the Tessa, you know, his co-workers did this mutation, and they were analyzing on SDS page yields how heavily modified the protein was by glycosylation. Because one glycosylation always occurred, that is this G1 glycosylation, because it's in the ER lumen, but the second glycosylation can only occur when the helix becomes not part of the bilayer, but becomes a soluble helix. And so by this, the other glycosylation site can be also recognized by the OST complex and modified. Very simple biochemical assay, something to measure then on SDS page and doing densitometry, how much is glycosylated or not so ever. So what he came up with was then with a simple experiment, and I like the simplicity of the experiment. Yeah, the simplicity of an experiment, but the complexity of the kind of conclusions that one can come up with. So this is here the kind of model helix they had here, and the kind of amino acids that they checked. And isoleucine, are the most preferred ones here, and then changed it, and got some profiles for all this kind of amino acids. How far away from the membrane center they want to be, in order to being incorporatable. And I think, that is some of the fancy things here, that you can also use hard biochemical work to get to some real, very interesting conclusions. So, and with this kind of story, oh, I'm still one thing more. Why is the signal sequence always positive? Yes, it's the amino terminus, yes. Yeah, because of some charge interaction with the translocon itself. But please read this by yourself, yes. And the transmembrane helix predictor that I have said to you, yes, remember about that what we have just seen? Actually, that was actually one of the bases, to get the TMHMM actually validated and also quantified against the experiment, in addition to showing for distribution of amino acids in transmembrane heels or on the distribution level in a water octanol mixture, because the water octanol mixture is giving you not necessarily the best the best estimate for the position in the transmembrane heat. And with this one, I'm now really, really, really at the end with membrane proteins for today. And I thank you very much for your attention. And when we have some time, or you have some fun, questions.